class. Okay, so we went through how an action potential works. You've seen this graph, right? So what's happening here, right? This is our resting membrane potential, about negative 70 millivolts. This here would be where we have our stimulus, okay? So another neuron telling our neuron to fire, giving an excitatory postsynaptic potential, okay? So that brings us potentially, right, in this case, to our threshold of negative 55 millivolts. And then we have our depolarization. So this here is our depolarizing phase once we reach positive 30, right? So this is where our sodium channels open up, okay? Our sodium voltage gated channels open up. Sodium rushes into the cell, bringing us to about positive 30 millivolts. Then once we get to positive 30, our sodium voltage gated channels are inactivated by that polypeptide ball. And now our potassium voltage gated channels open up, okay? Now potassium rushes out of the cell, so now we're losing positive ions. So we're gonna go back toward our resting membrane potential. And remember, those potassium ions take a while to fully close. So we get this area here known as hyperpolarization. So we go below negative 70 millivolts for a short time. And then our sodium potassium pumps bring us back to our resting membrane potential until we have another signal. So we have something else going on. We have two different phases where we can't really have another action potential happening until some things end, okay? So we have an absolute refractory period. Okay, and then we have a relative refractory period. So I'll tell you where each of those happens. Okay, so absolute refractory period is absolutely nothing could cause another action potential to occur. Okay, so that happens from where we start our action potential to our top here once we get to positive 30, okay? So from here to here, this is our absolute refractory period, okay? So no matter how strong the next signal is, nothing can cause another action potential here. Now our relative refractory period is where if we just get a normal strength signal that caused this action potential, for example, it won't cause another action potential. We would need a much stronger signal to overcome our relative refractory period, okay? And that's from here, right, at our positive 30, back until we get to our resting membrane potential. Now, normally, for a normal signal to come along again, we'd have to wait until we get to our resting membrane potential. So this here would be our relative refractory period from here to here, okay? So again, we need something very strong telling the muscle or whatever effector to fire again. Okay. Okay. So how does it determine how quickly the message gets sent through the neuron? Okay. So we have conduction velocity. That's the speed at which an action potential is sent through the axon length, okay? So two things really affect this. So it's gonna be the diameter of the axon. So larger diameter fibers have less resistance, so flow can happen a lot quicker, so faster impulse conduction. And then we have degree of myelination. So the more myelinated an axon is, right, the faster that information is going to be sent. Because remember, the action potential can skip those myelination and just skip to the myelin sheath gaps or nodes of Ranvier, okay? Okay, so we're gonna talk about one homeostatic imbalance with myelination. So you've probably heard of MS, multiple sclerosis, okay? And it's in the name, right? Multiple sclerosis means there's multiple like kind of scarring on the myelin sheaths. Okay, so it's an autoimmune disease, so your own body actually attacks the myelin on your nerves. Okay, so when that happens, they can get destroyed and they turn into these hard lesions known as scleroses, right? Hence the name multiple sclerosis. So when that happens, right, you're damaging the myelin. So now the conduction speed is going to slow down, okay? So you're not gonna be able to send messages as quickly to your effectors, okay? So the body does try to make up for this by adding more sodium voltage gated channels along the axon in areas where there's damaged myelin, okay? So your body can do that, which causes a remission, okay? So maybe you're feeling a little bit better, you're able to move a bit better, but then there's relapses as well because your body is still continuing to attack the myelin, okay, in other areas of the axon, okay? So vision symptoms include disturbances in your vision, weakness, loss of muscle control is a big one, probably what you think of when you think of MS, speech disturbance, and incontinence. So remember, we have those levator ani muscles, those pelvic floor muscles, so it could affect incontinence if the nerves to those muscles are affected. Okay. So treatment would be drugs that stop immune activity. Okay. So what is a synapse? A synapse is just a space. Okay, so synapse is a space between one neuron and another neuron, or a neuron and an effector cell. Okay. So we have electrical synapses and chemical synapses. So remember, we said that signals are sent both electrically and chemically through our neurons. Okay. So with an electrical synapse, there are gap junctions that connect the cells, okay? And it allows information to be synchronized throughout a group of cells, okay? So it would happen like in the heart, for example, and in the muscles, where you want things to happen all at the same time, okay? Chemical synapse, this is a one-way transfer, right? From a presynaptic membrane to a postsynaptic membrane, okay? So the presynaptic membrane would be the end of the axon terminal, the synaptic end bulb, and the postsynaptic membrane would be whatever it's sending a signal to. So another neuron, maybe dendrites on another neuron, or a muscle, or a gland, okay? So, and then a chemical is released, like when we talked about muscles, right? Acetylcholine was released, so that would be a chemical synapse. A chemical is being sent, okay, to the postsynaptic membrane, or to the muscle in that case, okay? Okay, we have three different types of synapses between neurons, so neuron to neuron, okay? Axodendritic, this is probably the most common, so this is where the end bulb of one axon communicates with the dendrites of the next neuron. Axoaxonic, the axon of one neuron, or the end bulbs, can communicate directly with the axon of another neuron. And axosomatic, so the soma, the cell body, okay? So the end bulbs of one neuron can communicate with the soma of another neuron. So those are just three ways that the axons can communicate with each other, okay? Okay, so how is information sent across a chemical synapse? Let's talk about the steps. I'm going to do my best to draw this out. So I'm just going to draw a little synaptic end bulb here, okay? And it's communicating with something else. So probably this, so this would be our presynaptic membrane, and this here would be our postsynaptic membrane, okay? This is communicating with this, presynaptic to postsynaptic, okay? All right, so what are those 6 steps? Okay, first, an action potential has to arrive at the axon terminal. Okay, so we went through how an action potential occurs. Okay, all that has to happen, and the action potential arrives this axon terminal, okay? Okay, then we have voltage-gated calcium channels, okay? Those are going to open up and calcium can enter into the axon terminal, okay? So it flows into the cell. So we have these right here, here. So from the extracellular fluid, or just the fluid surrounding here, we would have calcium entering into the synaptic end bulb. And obviously a lot of these would open up, and I'm just showing you two, okay? Okay. So now we have, in here we have synaptic vesicles. So it's just these vesicles. We know what a vesicle of a cell is, right? It's just something that holds something else. So these little vesicles, they're holding some sort of neurotransmitter, right? This is very similar to how muscle contraction action potential worked, right? So we have these synaptic vesicles. They're holding some sort of neurotransmitter, okay? Calcium causes these proteins here to attach to proteins on the vesicle. They kind of intertwine and they get pulled to the synaptic end bulb. Okay, then they open up and release, these are holding neurotransmitter, they release their neurotransmitter to the synaptic cleft, this space here, okay? Okay, so neurotransmitter is then released because of that calcium, okay? Then neurotransmitter is going to diffuse across this space here, right? So we have lots of our vesicles here releasing neurotransmitter, okay? And now it's being released into this synaptic cleft here, okay? So the space between our presynaptic and our postsynaptic vesicle, not vesicle, membrane, okay? So now there's these ion channels here on our postsynaptic membrane, whatever it is, a muscle, another neuron, whatever it is, okay? These neurotransmitter bind and open up these channels, okay? And that creates a graded potential, right? So now ions can enter in, okay? And it causes that graded potential. So it causes us, if this is an excitatory neurotransmitter, it causes us to go toward our -55 threshold, okay? If it's inhibitory, then it would cause us to go back down toward our resting membrane potential, okay? So the signal is sent, basically, okay? And now there's three ways. So now we can't keep this neurotransmitter here in the synaptic cleft, or else it'll keep these channels open and it'll constantly just be causing these graded potentials to occur, right? So three ways that these neurotransmitters are taken care of. Reuptake, okay? So astrocytes can actually take care of these and kind of take them away, or they could be taken back by the axon terminal. Degradation, so whatever enzyme degrades this specific neurotransmitter, whatever it may be, like remember with acetylcholine, there was acetylcholinesterase, right? The enzyme that degraded acetylcholine during the muscle action potential, okay? So an enzyme can come and take care of these, degrade them, or diffusion, they can just diffuse away into the extracellular space, okay? So make sure you know those three ways that a neurotransmitter effects can be terminated. Okay, so what happens in the synapse here takes time, okay? So there's time that's needed for the neurotransmitter to be released, to bind to receptors, right? So there is a delay from about 0.3 to 5 milliseconds. So you wouldn't notice it yourself, right? But there is a delay there, okay? So this is like the rate limiting step of a neurotransmission. This is what takes the longest and would cause the action potential to kind of be slowed down, okay? So the signal moving down the axon is very quick. This is what takes the longest time, is that neurotransmitter being released and binding to the channels on the postsynaptic membrane, okay? Again, not noticeable by yourself. You wouldn't notice that even 5 millisecond time delay, right? Okay, so that was chemical synapses. Electrical synapses, these are less common. And in electrical synapses, the neurons are electrically coupled. So again, they're joined by those gap junctions that connect the cytoplasm of the neurons that are near each other, okay? So communication is very, very fast, okay? So this is like found in brain regions responsible for movement of the eye, okay? Or in the hippocampus, which is like, we'll learn about the hippocampus, but learning and memory functions, okay? And also electrical synapses are very abundant in embryonic nervous tissue where lots of things are happening, right? You're creating a whole baby, okay? Okay, so we talked about excitatory postsynaptic potentials and inhibitory postsynaptic potentials. So again, we have excitatory neurotransmitters and inhibitory ones. Excitatory bring us toward our threshold, inhibitory bring us away from our threshold and back toward our resting membrane potential, okay? Okay, so how do we reach our threshold of negative 55, okay? So we have two different types of summation, okay, or how signals add up to that potential of about negative 55, okay? So we have temporal summation. This is when one or more neurons send signals in rapid fire order, okay? So it sends one signal, another signal, etc. really, really quickly, okay? That first impulse, let's say it produces a excitatory postsynaptic potential. So let's say we're starting out at negative 70. So it produces an excitatory, and then another one comes soon after, and it just kind of adds up, and then eventually we get to our action potential. Okay, if it adds up enough, if there's enough signals fired. Okay, that's temporal summation. Then we have spatial summation. So this is just when a lot of neurons are communicating with the same neuron at the same time, okay? So there's many receptors that are activated, okay? So that would just cause, that would be like this. Just one big leap, action potential, done, okay? Because a lot are communicating at the same exact time, okay? So they add up very quickly at the same time. Okay, so I'm just going to talk about a few select neurotransmitters and their function. So acetylcholine we already talked about with muscle action potential. Okay, so this is found in the central nervous system and peripheral nervous system. Okay, so this is an excitatory neurotransmitter. Norepinephrine and epinephrine, also known as noradrenaline and adrenaline, these are both excitatory as well. Okay, so involved in like attention and consciousness. Okay, serotonin. Okay, so this is important in like our emotional states. You've probably heard about this maybe in like psychology class. Okay, glutamate. This is helpful in memory and learning. Okay, and then we have GABA. Okay, gamma aminobutyric acid, GABA. This one's an inhibitory neurotransmitter, so this would move us away from our negative 55 millivolt threshold. Okay. So neurons, can they repair themselves? Can they regenerate? Not really, okay? So the nervous system exhibits plasticity, right? So it's able to we're able to like get smarter and learn, right? But neurons actually have a limited ability to regenerate themselves. They don't really regenerate themselves, okay? So regenerate is the ability to repair or to replicate, okay? So in the central nervous system, there's really little to no repair, okay? And this is because the neuroglia stop repair, okay, especially oligodendrocytes. During fetal development, of course, there's lots of regeneration, right? But after that, there's really no more cues that were present in the fetus. Okay, so that doesn't happen anymore. And scar tissue readily forms in nervous tissue, which scar tissue is not great for sending information. Okay, it kind of pauses that. So we don't want to cause a lot of scar tissue in our nervous system, of course. Okay, in the peripheral nervous system, repair is possible. It's still not very common, but it is possible. Okay, if the cell body is intact and the Schwann cells are functional. Okay, and then if we can avoid scar tissue from forming too rapidly. Okay, so what would happen is, throw one more thing out. So how it would repair, I'm just gonna draw my axon here with my myelin sheaths. Okay, so this is a myelinated axon. In pink here are my myelin. Okay, so if there's some sort of damage where the membrane kind of disintegrates here, okay, the Schwann cell can come and cause like this regeneration tube. So it can just kind of fix the area here, and then you would have myelination throughout. So this whole area would now have myelin. Okay, so that's how it just comes in and does some sort of limited repair there. Okay, but our cell body cells to be intact. But if there is damage to the axon, it can be somewhat fixed.